So yes, I will talk about deciphering the mechanisms of action for agricultural fertilizers. And see how do it right. So there is a gap, I feel, in research, especially when we think about research that occurs in academia, in universities, and then the application of it in agriculture, in the field. If you think about farmers and you think about yourself, that how much you know about how plants grow and how they respond to the environment, farmers have more of their experiential knowledge, not as much technical knowledge. And we don't have the experiential knowledge that they have, but we have the technical knowledge. So this gap is there because of this particular reason. This is my thought. In universities and institutions, let me see if I can use a pointer. Probably not. But in universities and institutions, research is done in a very different way. The research is done with a goal to generate intellectual property. There is looking for research funding, train students, and there is pretty much a professional ladder. You join a profession and you have to perform to advance, and with job security. Of course, you have to do publications, you have industry partners sometimes, and you have job security. So these are the goals in a university environment. Whereas if you look at agriculture and manufacturing, even fertilizer manufacturing, the goals are very different. These companies have to sell. They want high yield. They have to increase their sales. They want to reach bigger markets. They have annual revenues, get into pipelines, and above all, they have competition. They have other products of similar type. So in universities or academia, it's publicly funded. The money comes from usually from government agencies, taxpayers' money, and the any technology that is developed is owned by universities. You all know this. I'm just repeating just so that we can highlight the problem. Whereas in agriculture, it's privately funded usually, where the money has to be returned, revenues have to be generated, and there is market value. So the gap is there because these two pillars, these two different sectors don't communicate very well with each other. And the reason is that the universities have knowledge, and if any knowledge needs to be transferred or translated, it has to go through intellectual property. Then there is investments, and those investments require returns. And agriculture in general doesn't have very large returns like pharmaceuticals do. So this pharmaceutical industry is a good complementary example, but agricultural industry is lagging behind in science. I joke about it. It's not really a joke, but it's almost like going to a hospital and all the doctors are developing new drugs, and the only thing you have access to is the clinic. That's where agriculture is, because all the doctors are developing their own products, and none of those doctors are communicating or connecting with the agriculture industry. Okay, so how do we change that? Well, first we have to understand the problem. What is the real problem that exists in this area? The real problem is that farmers, they want to grow and they want to make money, but they get every day, they will get so many. I'm from California, and California is the bread and butter of the entire US. We say that because 60% of all the specialty crops are grown in California for the entire United States. This is a fact. So California is a very productive land, and it's pretty long and big, and there's a lot of agriculture. When I say specialty crops, I mean not corn, not soybean, not canola, all the food that we eat, tomatoes, onions, beans, peas, all of this is grown in California. So in California, there are many companies that will come to farmers and they'll say, well, look at this picture. If you use my product, your tomato will be 3 times as big. And it's just a photograph, right? And farmers get this 10 times a week, at least. They get so many salespeople coming to them, buy my product, buy my product. I talked to one farmer. He said, now, how is it possible that every single product is better than the other one? How is that possible? Because that's what everyone is telling them. My product is better than the other one. So how do they choose? Well, there is no science. There's no mode of action. So because of this reason, in 2015, I started this company, i-Cultiver. i-Cultiver stands for us making an effort to cultivate new relationships between agricultural farmers, growers, and academia. And so the way to do that, I thought, was to not make it reliant on investments. So i-Cultiver has never raised a single dollar. And I will show you how it works. And I will invite all of you if you like this idea, I would invite all of you to connect with me, and we can see we can do some projects. I am going to talk about quite a bit of science. I wasn't sure the audience here, since the title was Plant Molecular Biology, I'm going to show you a lot of molecular biology. So the way I started i-Cultiver was to do this deep science of molecular biology, to do mode of action, but to do it in collaboration. So I'm from Carnegie Institution of Science, which is Andrew Carnegie Foundation funds the Carnegie Institution, which runs the plant biology department at Stanford University. So I'm employed by Carnegie Institution, but we are on Stanford campus, and we have a close collaboration with Stanford University. So my colleagues most of them are from the Bay Area. Dr. Wang is at the Carnegie Institution. Dr. Katie Krorikowski teaches at a community college. Dr. Bogomolny is from UC Santa Cruz. So I'm just going to show you. So you can see that it's a good mix. We have Dr. Kent McHugh, he's from the United States Department of Agriculture. And so each person brings a different knowledge and experience. So in this collaboration, in this consortium, we can get most of the agricultural products studied by students. So I'm going to show you how all of this works. So what was needed after this consortium was created was students. I wanted to do it through students, so that not only that we help and answer some questions about how products work. Perfect. Okay, so because we had the consortium, now we have to get the work done. And I wanted to not use, in a way, it comes to mind because, students don't get paid that much, but not use students, but engage students to perform these research projects, so that we also train the next generation of researchers while we are looking at agricultural products. So these are different places that the BEST program, which is known for biotechnology education and specialized training. And we do field trials, greenhouse trials, we do pretty much anything that a lab can do, and also in the field. And we have different groups of students each year. It's an annual program, and it's a paid annual program, so they get paid. But we have, as you can see, we have access to greenhouse. That field in the middle on top is at Stanford in the back. So we grow crops over there. And so these students are really great, and they do really hard work. So this is the website, i-Cultiver. And you can see that we provide basically research services. So the companies, I'm going to talk about these two companies, Ezomite and Y-Design, the research that we've done for them. So the companies fund the projects, and the research is done completely independently. The goal is to publish. So just similarly as in university, the only difference is that the company provides the financial support for the students, and the work is done at universities independently for the publications. And once the publications are done, these companies gain access to high-end research, and with publications, then they don't have to just go and show photographs. They actually have mode of action. So I'm going to tell you about this research. We'll start with Ezomite. Ezomite is a company that sells volcanic ash that a surface mine from Utah. It was a volcano that erupted about 3 million years ago. And there's a huge mound. I think they say that they have enough for 300 years of volcanic ash. They grind it into different rocks and powder form, and then they sell it for agricultural inputs. They've been doing this for 20 years, but they were one of the first companies who actually wanted to know how does this volcanic ash help crops. So we started working with them about 4 or 5 years ago. And this was in collaboration with Dr. Britt Koskela, and she's at UC Berkeley. And we always engage the head of research of the company itself, because if the company has any research internally, it's very important to know what they have done. So John Farrow is from Azomite. He leads their research efforts. So the three of us started looking at what we could do first. So we started with looking at tomatoes. We added the volcanic ash to the soil and grew a bunch of tomatoes in the greenhouse. And we found, as you can see, these are weeks on the x-axis, going from weeks 9 to 22. The first week, both the treated plants, which are in red, and the control plants, untreated, which are in blue, both of them gave pretty much similar tomatoes, which were not very, very much per plant. So we are looking at tomatoes on plant per week. And then this is per plant per week. So as we look at harvest over the weeks, there is a clear increase in tomatoes, total number of tomatoes that were harvested over the process of the entire growth period with treatment of with Azomite treatment. So we always do this first so that we don't start looking at products that don't have an effect. So we always do our own research either in the greenhouse or fields before we move the product on to further deeper research. So we were convinced that there is something here. So we grew these tomatoes and we collected rhizosphere, root, and phyllosphere and endosphere for determining which bacteria, which microbes were present. So first thought was, because we're adding volcanic ash to the soil, perhaps it's acting through different microbial communities. So we started to look at the species that were present in the soil and inside the plants. What we found was very interesting, especially in the endosphere. So rhizosphere is the the soil that's on the surface of the roots. Endosphere is actually inside the roots. And what we found that inside the roots, in the endosphere, the microbial communities contain more species that use complex carbohydrates, like sugars, like C6, the carbohydrates that have 6 carbons. Whereas the untreated, without Azomite, the microbes that were there, they used 2 or 3 carbon carbohydrates, or 2 or 3 carbon compounds. So there was a clear shift in microbes that were consuming sugars inside the roots when we had Azomite. So that means the plant was making more sugar. How does a plant make sugar? Photosynthesis. So it was one of the first clues that perhaps Azomite somehow is helping the plant increase photosynthesis. That was our first thought. But this was published, and we started to look in that direction. So this basically shows that with Azomite, there was an increase in carbohydrate volume inside the roots. So we had more microbes consuming that. So, but that's endosphere. I will also touch upon phyllosphere. Phyllosphere, which is shown up there, which is basically above ground, all the microbes that live above ground. Okay, so we'll talk about that in a minute. So after that clue, we started to look at photosynthesis. And so the first study that was designed was with Dr. Kent McHugh and again John. And Dr. Kent McHugh is at USDA. So what we decided to do was to see whether Azomite actually affects photosynthesis. And what is an easy way to test that? Easy way to test that is to see if Azomite's effect is dependent on photosynthetically active radiation or light. So this is a very typical, I'm a photobiologist, so I love doing these kinds of analyses. These are basically 4-day-old seedlings of Arabidopsis. And you can see that on the top they're grown in continuous dark, in the middle they're grown in the red light continuously, and in white light. So what we can see that in the dark, they're etiolated, so they grow very tall. In red light, they open their cotyledons and start to green, and so they do the same in the white light. The difference in white light and red light is that there is blue and red in white light, the two photosynthetically active radiations. In red, it is just red. So because it is just red, they are still slightly more elongated. but we can clearly see that both in white light and in red light, they are more, they're growing a little bit taller. You can see the numbers below because it's blocked here, but it says 0, I think it says 0, 0.1, 0.5, and 1. So that is the grams per liter. So when we add 0.1 and 0.5 grams per liter of rhizomite to the media, growth media, we can see that these seedlings are growing bigger. Whereas when we go to 1 gram, it is actually a negative effect. It appears they're growing smaller, shorter. So that was interesting. So we then looked further and did a more elaborate analysis. So you can see we grow the seedlings in the dark, in red light, and in far red light. Now, far red light is light, but it is not photosynthetically active radiation. For photosynthesis to occur, plants require red and blue, not far red. Far red is more like an evening light. So in far red, they don't even green. And so we clearly saw that in far red, the effect of rhizomite was not present, whereas in red, they grew slightly taller. And this is significant. This assay is very well established. So even a few millimeters is very consistent. And this is point measure. We can measure the length of the hypoportal. I'm not sure if I'm showing that next. We will see. But this aligned with what we were thinking. Of course, we tried phytochrome A and phytochrome B mutants. Phytochrome is a photoreceptor. Phytochrome B perceives the red light, and phytochrome A perceives far red light. You can see that with phytochrome B mutant missing in the red light, the seedlings grow very tall, whereas with phytochrome A missing, seedlings grow very tall in far red light because those photoreceptors perceive those specific wavelengths. So this then showed. Yes, I am showing it. So that's good. This is just a summary of what I just showed. We can see that when we add azomite, so you can see in white and in red, right? So the white portion is showing white light and red light, and the different amounts, like 0.1 grams per liter, 0.5 grams per liter. You can see that both 0.5 grams per liter increase both cotyledon area, which is on this side, and hypoportal length, which is on that side. So if we go above the center line, which is the control, if we go this way, we are increasing hypoportal length. If we go this way, we're increasing cotyledon area. So which means if you're on this side, we have increased growth, right? So with 0.5 and 0.5 grams per liter under white light and red light, we got increased growth. But when we increase to 1 gram per liter, that impacted growth negatively. This is really important. So when growers are adding a product like this, if they use too much, it can actually damage their productivity, right? So it's important to study the amounts that we need to apply in agriculture as well. So from this study, then it was telling us that azomite was increasing growth, but it required somehow photosynthetically active radiation, red or blue light needs to be there, or white light. Well, of course, sunlight will have all of those colors. So now we thought, okay, we have an effect on microbiome, endosphere microbiome. What about the phyllosphere? So another thing that i-Cultiver has been doing in the past 4 or 5 years, which has been really fun, because we initially started taking projects from industry and then engaging students to do research. Some of the professors or some of the other faculty around us in the Bay Area who develop new products, they file intellectual property. And what does normally happen? You file an intellectual property with your university, and the university will say, oh, great, now we own this. We will find someone who wants to buy it. And that's it. If nothing else happens to it, unless if there is, the universities are not very good in advertising, unless if some publication attracts like Monsanto or Bayer's attention, that does not go anywhere. So now we have started also to help PIs bring their IP. PIs bring their IP out into commerce. So what we do is we take projects. This is one of the first projects that we started with. It's for a phyllosphere bacteria. I will talk about that. So Dr. Britt Costello developed 16 strains of microbes, bacteria that she collected from tomatoes, and she's isolated them, and we call that phyllostock. phyllo for above ground, start for applying bacteria, and the start of growth of seedlings. So PhyloStart is a new product that Dr. Britt Costella had an IP on, and i-Cultiver has now licensed it from UC Berkeley. So we have taken over the project from UC Berkeley by license so that we can actually commercialize Dr. Britt Costella's IP. This is a rare thing because usually the PIs have to start their own companies to do that. So what we decided to do was to combine the effect of phylosphere with azomite, because we already had azomite, and this is PhyloStart. So you can see the PhyloStart bacteria that are established on leaves. So on the left are control plants, and by the way, these plants were randomly grown in a greenhouse. They were close to each other. When we apply PhyloStart, we move them away from the other plants. After application, they go back. So the bacteria are not jumping, or they're not mixing with the control plants. But these are all the PhyloStart bacteria. All 16 of them are shown over here, and they're not present in large amounts on control plants. So this was really interesting when we did this experiment, and this took a long time to do. So this is showing tomatoes produced per plant, and you can see on top how much azomite was added: 0.512 gram. We went up to 3 grams because Arabidopsis is a much smaller plant and tomatoes are bigger. And we wanted to find a range where we saw again the negative effect of too much azomite. So if you just look at the blue bars, you can see that just with azomite, which is the blue, from control to 0.5 grams, 1 gram, 2 grams, we have a consistent increase in productivity. When we get to 3 grams, we have a drop in tomato productivity just with azomite, right? Similar to what we saw with the Arabidopsis. Now, if we apply PhyloStart, PhyloStart is the pink one or red, the pink color there. When we apply PhyloStart just on control, so in the first one, there is no azomite, it's only PhyloStart. We can see that with PhyloStart, there is a significant increase on its own, similar to just azomite 0.5 grams. They're all there. But when we add PhyloStart and azomite, we have a synergistic effect. This was a surprise. So PhyloStart is being added on the leaves, and azomite is added in the soil, and we almost doubled the production of tomatoes in the greenhouse with 2 grams of azomite and PhyloStart. Not only that, when we saw a drag or a dip in the production with azomite, when the plants had PhyloStart on the leaves, we lost that dip, and it produced similar to the control as if it wasn't treated. So this was a really interesting aspect of looking at how microbes can help plants deal with stress at the end, and also microbes and nutrient inputs like azomite, which is micronutrients, minerals, trace elements, how they can interact with each other. This study was also very interesting, and actually this became quite popular. It was picked up by the local newspapers because what we showed here is we applied Pseudomonas syringae, which is a pathogen, as you know. So Pseudomonas syringae causes disease on tomatoes. In the first part here is autoclaved soil, so it doesn't have any microbes. Second one is also autoclaved soil, but in the second one we added organic phosphorus, right? So it has fertilizer. So the first side on the left is without fertilizer, autoclaved soil, no microbes. Right side is autoclaved soil, no microbes, but with organic phosphorus. What you're seeing is the blue, which is Pseudomonas syringae, on causing disease on tomatoes. But when we add PhyloStart, which is the pink, we prevent it. We were able to suppress the establishment of Pseudomonas syringae with PhyloStart bacteria, but that did not work when there was too much fertilizer. So the only interpretation of this result is that the way PhyloStart, or I would say any bacteria that provide disease tolerance, is possibly through competition for nutrients, for food. So you're competing for food. But when you provide too much fertilizer, there is no competition. That allows both the pathogen as well as the natural microbes to grow equally. So this was interesting and quite revealing that too much use of fertilizer, as we've seen not only with azomite, but also now with organic phosphate, that using too much fertilizer can be actually a bad thing. So i-Cultiver took over, like I said, of Phylostar, and just earlier this year we registered it. This takes quite a bit of work. I don't know if many times companies will spend $7 to $10 million to get their products registered. We did it in like less than $200,000 and in like 12 months. So this product is now registered with the state of California, and it is available for commercial sale. And now we are again developing a whole group of students who are going to be scale manufacturing it. So that we are going to scale it up again with the help of students. Okay, now we're going to talk about the very last part of azomite, and then a few slides on our design, and then I'll be done. So because of all the results we had seen, it was a really good clue that if azomite is working through photosynthesis and it's interacting with microbes, there has to be some effect on plant gene expression. So it had to be done. So we did a RNA sequencing gene expression study using an arabidopsis. We used similar growth system with the seedling that I showed you earlier, but I also used phytochrome B mutant because we saw an interaction with photosynthetic radiation, and phytochrome B is a red light photoreceptor. What we did was to compare the effect of azomite application in Colombia, which is the wild type of arabidopsis, as well as the phytochrome B mutant. So we applied azomite on both, and we looked at the differences between the genes that are regulated by phytochrome B itself without azomite, and genes that may be regulated by azomite in Colombia, the wild type. So this shows the overlap. These are all the genes that were regulated, that are known to be regulated, and we also found that again by the phytochrome B mutant. When the phytochrome B is mutated, we can see genes misexpressed, going down in like blue means they are suppressed, going up in red means they are increased in expression. So we can see that all the genes shown here are targets of phytochrome B that are misexpressed in the phytochrome B mutant. But these are also the genes that are regulated or impacted by azomite application. Sometimes they are affected to the same direction. So azomite either increase or suppress some of those genes, whereas phytochrome B also regulates those genes, and many times in the same direction, sometimes in the opposite direction, almost actually 50-50. These are all the genes that we discovered that are regulated by azomite. On the left are phytochrome B dependent, which means that they require phytochrome B for azomite to act. And on the right are phytochrome B independent, so they did not require phytochrome B activity. For some reason, it's a very good reason, and I can elaborate on that if you ask me a question after this. We picked Superman. If you have not heard of Superman, Superman is a gene that became quite popular. Obviously, all of us who work in labs, we get bored, so we come up with really cool names. Superman is a gene that is a master regulator, transcription factor, which regulates the deposition of suberin. Suberin is a wax compound. So Superman regulates other transcription factors downstream to deposit wax in the roots. And that was suppressed in response to azomite application, which was quite interesting. So we actually got Superman mutant, which is the submutant. And with the submutant, obviously that gene is not active, the transcription factor is not active. So the question was, if we apply azomite to submutant, it should not, it should be actually more sensitive, because if azomite is suppressing Superman and we take submutant, which doesn't have Superman, it should be more sensitive to azomite. And it is. That is some data I'm not showing yet, but we have done that research. I wanted to show you even more interesting. Superman is a wax compound which we can stain and we can detect through confocal microscopy, like scanning microscopy. And so these are images. You can see Colombia, which is the wild type, It's very hard to see, but I think we don't need to do this. But if you were to turn the lights down, you'll see a very faint blue light. That is the same suberin on the outside of the roots. When we add azomite to Columbia, this is 40% increased brightness. So there is about 40% reduction in suberin deposition in Arabidopsis. And when we look at the Suberman mutant, we don't see any of that. So this confirmed that with application of azomite, we at least have about 40% reduction in suberin deposition. And so how does this make sense? Why is the wax being affected? Well, our hypothesis, which we are currently working on, is that when we add azomite like a volcanic ash, which has over 70 different micronutrients, the plant responds to it by suppressing its wax deposition. Wax in the roots is usually kept by plants to retain more water, especially around drought. The plant will not lose water, but it also prevents uptake of nutrients. So the plant will reduce its wax deposition so that it can take up more of those micronutrients, minerals, and we think that some of them are like magnesium, manganese. These will go up and increase chlorophyll and increase photosynthesis and leading to more sugar production. That sugar goes down to the roots of the seedlings, which then recruits other wonderful microbes from the surrounding areas. So in a whole, this suppression of wax allows the plant to take up more nutrients. So that is our current mode of action for azomite. And we can see hints to that. This is just a KEGG function analysis. From the gene expression study, we looked at which functional sets of genes were affected. And we can see that carbon fixation, photosynthesis is increased on the top. And the ones that are suppressed are cutane and suberin and wax deposition. So this aligns with what we are thinking, but of course, more work is needed to complete this work. So, okay. So we'll skip this because I've already covered that. This just talks about, about the mode of action. I'll cover the second one. It's only a few more slides. Vitazyme. Vitazyme is another product that a company sells, and we just developed this manuscript. It's just been submitted. But Vitazyme is a phytohormone-based product. They claim on their label that they have steroids, and so we try to help them to show them how their product works. The first study we did was with specialty crops. We had kind of increase in total overall production. Some of them were significant, like broccoli and tomatoes, but there was an overall increase. So we picked this up as a product as well to study, and I'm showing the label. It says it has homobracinolide. Bracinolides are steroids. They're plant hormones. The problem was that the company did not have any means to detect homobracinolide. So with Dr. Roberto Bovamolni from UC Santa Cruz, we developed this spectrophlorometric assay where we use fluorescing compound, which is known as GABA, danzylamino phenyl boronic acid, that has a fluorescence, and it binds specifically to all brachiostenoids. So with that, we could use a spectrophlorometer to detect fluorescence, and so we can do excitation and emission, and we can see that with the controls, bracinolide, these are controls, which means standards bought from Sigma. So we can look at bracinolide and homobracinolide, and we can see specific peaks. You can see peaks at 505 and 342 for excitation and 505 for emission. When we do Vitazyme, so that's Vitazyme, we see the exact same peaks with the spectrophlorometer. So now it's a very simple fluorometric assay, because outside of this, the only other way to study these very low amounts of steroid is using mass spec, HPLC or mass spec. We can see that there is an increase as we add spike with more steroids. We can see a linear trend, so we know we are looking at the right thing. We did go to mass spec. We have that at Stanford. So by mass spec, we were able to detect homobracinolide. There's a standard of homobracinolide, HPL, and then blank, and then this is Vitazyme. So it does indeed contain homobrassonolite, as the company suggested. And we quantified it. So these are 3 independent batches from Vitazyme, and we were able to see that it's about 1 micromolar. These steroids work at very low concentrations. We were also able to use a mutant, which is the DEECH mutant or deethylated. Then you can see in the dark that mutant is very short. The reason it is short is that it lacks the biosynthesis of brassinosteroids. But if we apply brassinosteroids, supplemented HPL homobrassonolite, we can rescue or recover that short phenotype, and we can do that with Vitazyme as well as the standard. So confirming that the homobrassonolite is actually active. So we showed that Vitazyme increased yield, contains homobrassonolite. We developed the spectrophlorometric assay, did the bioactivity assay. This is it. So just wanted to go through the last part quickly. I do have a podcast. As you can see, there's John Carroll there. We started this podcast to communicate the research that we do, and we invite students. And if you have a product or some cool stuff that you'd like to share on a podcast, you are welcome to contact me. The podcast is called Terra Science. So I'm showing an example of a podcast with Vitazyme. Earlier was with Azomite, and then the Phylostart. Actually, we had a chance to talk with Deepak Chopra, and so we've been on his channel as well, talking about Phylostart. That's the 16 bacteria that we apply on top. So we've been busy, but in a fun way. I'm not going to talk about the other aspect that I work on. Because I'm a photobiologist, I discovered, well, by planning that how plants bend towards light. I can block the bending or response to light by anesthetics. So if I give an anesthesia to seedlings that are bending towards light, if I give anesthesia, they stop or don't respond for 5 to 6 hours, and then they return. So it's a transient block of response to an environmental signal, very similar to what happens with anesthetics in animals and humans. It's nothing to do with plant consciousness. We are just trying to study how anesthetics work, because it is actually not known in animals and humans. So on Terra Science, we talk about that. And this is the very last slide. I hope that more of us can contribute towards agricultural understanding how products work in agriculture, because ultimately we all eat the same food.